The effects of genetic variants previously associated with fasting glucose and insulin in the diabetes prevention program.
Presented by Elizabeth Cain and Jessica Carter. This study was found at PLOS One and was published online on September 11th, 2012, with a reference of 10.1371/journal.pone.004424. This study was a longitudinal trial in association with the Diabetes Prevention Program, which investigated methods of diabetes prevention in high-risk groups.
These high-risk groups consisted of people with mutations in G6PC2, MTNR1B, GCK, DGKB, GCKR, ADCY5, MADD, CRY2, ADRA2A, FADS1, PROX1, SLC2A2, GLIS3, C2CD4B, IGF1 and IRS1 receptors, with a focus on MTNR1B and G6PC2.
MTNR1B stands for melatonin receptor 1B, which is usually involved in the maintenance of circadian rhythms in the retina and in the brain. MTNR1B has been associated in multiple studies with a 20% increase in the risk of developing type 2 diabetes.
G6PC2 stands for glucose-6-phosphatase catalyst 2, an enzyme involved in gluconeogenesis and glyconeogenesis.
The Diabetes Prevention Program was an experiment designed to investigate the effects of lifestyle change, a drug by the name of metformin or troglitazone, to prevent the onset of diabetes in high-risk groups. It was conducted in 27 US centers with 3,234 patients, randomized to each condition. However, the 585 participants randomized to troglitazone were terminated early. This was due to the drug being removed from the market after evidence of hepatotoxicity, damage to the liver.
In order to enroll for the study, patients had to have a fasting glucose between 95 to 125 milligrams per deciliter and an impaired glucose tolerance, 2 hours glucose between 140 to 199 milligrams per deciliter after a 75-gram oral glucose tolerance test. The endpoint of the experiment was diabetes incidence in the patient. Insulin resistance was calculated by 22.5 over the concentration of fasting insulin times the concentration of fasting glucose. Insulin secretion was calculated by the concentration of insulin at 30 times the concentration of insulin at 0, divided by the concentration of glucose at 30 minus the concentration of glucose at 0. The oral disposition index was calculated by 1 over the fasting insulin times the insulinogenic index.
In this experiment, genotyping was carried out. Where certain assays failed, proxies, or substitutes, were used. DNA was extracted from blood leukocytes. In this experiment, they used allele-specific primer extensions of multiplex amplified DNA products. They also used Cox proportional hazard models. These are used to model survival rates statistically.
In Table 2, you can see that the G6PC2 fasting glucose was decreased. The fasting insulin was increased. The insulin index was increased. The insulin sensitivity was decreased, as was the oral disposition.
In Table 2, you can also see that with MTNR1B, there was no change in the fasting glucose, the proinsulin adjusted for fasting insulin levels had increased, and the insulin index had decreased.
In Figure 1, Panel A shows the fasting glucose versus the genotype, and Panel B shows the insulinogenic index versus genotype. The fasting glucose was higher in carriers of the G risk allele, and the insulinogenic index was lower in these people also. Even after adjustment for the baseline levels, however, a significant improvement was seen in all of the genotypes.
In Table 4, you can see that gene DGKB had a significant reduction in fasting insulin by metformin, but no major changes by lifestyle. In GLIS3, fasting insulin was dramatically reduced by both the drug treatment and the lifestyle changes, and in MADD and C2CD4B, fasting glucose values were increased by both metformin and lifestyle treatment. Table 5 shows that for both G6PC2 and MTNR1B, diabetes incidence was high in the placebo group, but significantly reduced in both treatment groups for the associated alleles.
The loci do affect the fasting glucose and diabetes incidence at genome-wide significance. The authors believe that it is not the elevation of fasting glucose, but how it is raised that determines diabetes. They also proved that risk-related alleles at MTNR1B, GCK, and SLC30A8 confer a stronger rate of progression from normal glycemia to IFG than from IFG to type 2 diabetes. They have demonstrated that the three loci with the strongest reported effect on fasting glucose, MTNR1B, GCKR, and G6PC2, have consistent effects in the diabetes prevention program.
MTNR1B is shown to be associated with a reduced insulinogenic index. This gets worse over time, and genotyping it may be useful as an early detection. Early detection of this allele may lead to earlier diabetes prevention treatments. G6PC2 has conversely been shown to promote better beta cell function and have no major effect on the incidence of type 2 diabetes. There is no evidence of a non-additive reaction between the two.
The conclusions that can be drawn by this experiment are that diabetes onset can be delayed by drugs or lifestyle modifications.
It also shows that several previously associated genotypes may not have a causational relationship with diabetes.